Good morning, viewer. Bien this year. Oh, it's a breaking news. Yes. What makes us human? According to neurobiologists, it is our neocortex. This outer layer of the brain is rich in neurons and lets us do abstract thinking, create art, and speak complex languages. An international team led by Dr. Marike Albert at the Center for Regenerative Therapies restaurant of TUD Dresden University of Technology has identified a new factor that might have contributed to neurocortex expansion in humans. The study was published in the EMBO journal. Yeah, the neurocortex is the characteristic folded outer layer of the brain that resembles a walnut. It is responsible for higher cognitive functions. The neurocortex is the most recently evolved part of the brain, says Dr. Albert, research group leader at the CRTD. All mammals have a neurocortex, but it varies in size and complexity. Human and primate neurocortex have folds while, for example, mice have a completely smooth neurocortex without any creases. The folds characteristics of the human brain increase the surface area of the neurocortex. Yeah, the human neurocortex has a greater number of neurons that support complex cognitive functions. The molecular mechanism driving neurocortex evolution are still largely unknown, which genes are responsible for interspecies differences in neurocortex size. What factors have contributed to brain expansion in humans? Answering these questions is crucial to understanding human brain development and potentially addressing mental health disorder, explains Dr. Albert. To search for factors influencing brain expansion, the Albert group compared developing brains of mice and humans. Stem cells in mice don't divide as much as don't produce as many neurons compared to primates. Humans, on the other hand, have a large number of stem cells in the developing brain. This highly expanded pool of stem cells underlies the increase in number of neurons and brain size, explains Dr. Albert. The team found a factor that is present in humans but not in mice. Using 3D cell culture technology, the group tested if the newly identified factor could influence the expansion of the neurocortex. Thanks to the research awarded with the Nobel Prize in 2012, it is possible to turn any cell into a stem cell. Beautiful. Such a stem cell can be transformed into a three-dimensional tissue that resembles an organ, that is called a brain. Human stem cells make it possible to study development and diseases directly in human tissues, explains Dr. Albert. BN Das exclusive report.